Clathrin Mediated Endocytosis, also known as Receptor Mediated Endocytosis, is a process by which cell absorbs macromolecules, hormones and larger proteins inside of the cell with the help of clathrin, a protein that is present inside the inner leaflet of the plasma membrane. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about Clathrin Mediated Endocytosis, that is also known as Receptor Mediated Endocytosis. So what is the Receptor Mediated Endocytosis? So it is a process by which cell absorbs macromolecules, hormones and proteins through the invagination, that means pinching of the cell inside of the cell, and it get absorbed those molecules or proteins which can transfer from one side of the cell to another side of the cell on its own. That means if we consider this is a cell or membrane, that is a plasma membrane, so this is a plasma membrane, okay. So small molecules like gases and other small proper molecules that easily penetrates or comes throughout the plasma membrane, but large molecules which cannot pass through this plasma membrane are transferred from one side of the cell to another side of the cell by receptor, okay. So here receptor plays an important role for the transfer of one macromolecule from one side of the cell to the inside of the cell. So this is the whole process or mechanism of the receptor mediated endocytosis, in which a receptor plays an important role. So basically a receptor is a membrane bound structure. So if we consider this is a receptor and this receptor is a membrane bound structure, so basically it has three functional domains or parts. First of all, one is outside of the cell. So one is outside of the cell, that is called as extracellular domain. So extracellular domain. Another word is inside of the plasma membrane, and this portion is known as transmembrane. So transmembrane. And another part is inside the cytosol, and this portion is known as cytosolic domain. Cytosolic domain. So basically all three parts, that is extracellular part, transmembrane part and cytosolic domains, those three parts consist of a receptor, and these receptors are very much essential for the import of these particular molecules inside of the cell. One common example is the uptake of LDL, that is low density lipoprotein, into the cell. So low density lipoproteins are larger molecules, and the low density lipoproteins or LDLs cannot pass through the plasma membrane on its own. That is why it needs a receptor, that is a LDL receptor, which is comprised of 839 amino acid residue, and it has also three parts: the extracellular part, the transmembrane part and the cytosolic part. And if we look into this receptor in a closer manner, then we look or observe the modification of cytoplasmic domain. So here is the modification. So in the cytosolic domain, there is a 4 amino acids are present, and those 4 amino acids are asparagine, proline, X. That X can be any amino acid, and tyrosine. So basically asparagine, proline, any amino acid, that is X, and tyrosine, those 4 amino acids plays a crucial role to uptake or activate these LDL receptors. So whenever LDL or low density lipoproteins gets attached with this particular receptor, then the cytoplasmic domain, that means the 4 amino acid gets activated, and those 4 amino acid helps to accumulate these LDLs in the vesicle. That vesicle is ingested or invaginated by the cell, and the cell absorbs these LDL receptors. So another important proteins known as clathrin. So clathrin are basically a protein that is present inside of the inner leaflet of the cell or cell membrane. So basically these molecules are clathrin molecules or proteins, though they are present inside of the inner leaflet of the plasma membrane, and whenever these amino acids are getting activated, that means the C terminus of the receptor, LDL receptor, gets activated, then clathrins accumulates side of these particular receptors and creates a vesicle or vesicle like structure, and with the help of this vesicle these LDLs are getting absorbed by the cell. So this is the structure of clathrin. So clathrin is made up of two chains, that is, one is clathrin heavy chain, that is long one, long chain is clathrin heavy chain, and short one is a clathrin light chain, and another one is the terminal domain or of the clathrin heavy chain has a globular domain. So these domains plays an important role in the attachment with this particular LDL receptor. So what is the process? Now I'm going to explain the process by which these LDL or low density lipoproteins are getting absorbed by the cell. So if this is a plasma membrane, so there is a receptor present, okay. So this receptor is LDL receptor. So whenever LDL molecule or low density lipoprotein molecule comes in close contact with this particular LDL receptor, then the C-terminal domain is actually changed or getting activated, okay. So whenever this C-terminal domain is activated, and now you can observe there is a structural difference in the C-terminal domain, right? So there is a structural difference in the C-terminal domain. Now the clathrins are now accumulates inside of this plasma membrane or inner leaflet of the plasma membrane, but clathrin cannot work alone, they needs its adapter protein, and clathrin has 4 adapter proteins. So clathrin have 4 adapter proteins, that is AP1, AP2, AP3 and AP4. So these adapter proteins helps the clathrins to ingest or create a vesicle of this plasma membrane, okay. So here AP2, in this case, in the case of LDL uptake, there is a clathrin and AP2 complex plays a crucial role, okay, or vital role. So whenever these clathrins along with this AP2 or the adapter proteins are accumulates inside of the plasma membrane, then the plasma membrane invaginate itself and creates a vesicle like structure like that, okay. So here is the receptor and LDL is present along with this receptor, okay. So whenever this clathrins creates a vesicle, then it accumulates inside the inner leaflet of the plasma membrane, okay. But here is the problem, here comes the main problem, that it cannot pinch itself by the help of clathrin. That means the vesicle cannot pinch it from the plasma membrane and create a compact vesicle. For this reason, another protein known as dynamine, so dynamine comes into play. So basically dynamine are arranged or accumulates in the neck region of these growing vesicles. So here dynamines are accumulates in the neck region, so these proteins are dynamines. So whenever dynamine accumulates in the neck region of this growing vesicle, then this dynamine hydrolyzes, so it hydrolyzes GTP, and by hydrolyzing GTP it creates or produces energy. So it produces energy. Whenever the energy is generated, then pinching off the vesicle pinching of the vesicle occurs, okay. So whenever these dynamine are accumulates in the neck region of these growing vesicles, then the pinching off comes into play, and it pinched off, that means the growing vesicle now pinch off from this plasma membrane or surface of the plasma membrane. So now this plasma membrane along with the receptor and LDL creates a vesicle, okay, and this creates early endosome, okay, early endosome. So in case of the early endosome, now the endosome is getting inside of the cell and the pH changes occur. So whenever pH change occurs, that means from high pH to low pH, then the receptor gets separated from this LDL. So now receptor is separated from the LDL, so it is the LDL. So LDL is now separated from the receptor, and these receptors are going back to the plasma membrane. That means it recycles itself, and this LDL comes inside the cell and absorbed by the cell. So this is the LDL, and now this LDL absorbed by the cell, okay. So this is the whole process of receptor mediated endocytosis, how clathrin is worked. And another important protein that also helps in receptor mediated endocytosis known as caveolin, okay, caveoli. So clathrins and caveoli, both of the proteins works in receptor mediated endocytosis, but in case of caveoli, here caveoli also creates a vesicle, and the structure of caveoli slight difference from the clathrin, and this is the triangular like structure, and here also dynamine plays an important role or crucial role. So caveoli also helps in receptor mediated endocytosis along with clathrin. So this is the whole process of clathrin mediated endocytosis along with the also known as receptor mediated endocytosis. I hope you like that video. If you like that video, please hit the like button. Don't forget to subscribe, and thanks for watching.